Stefan Ropola is the co-moderator. I'd like to invite James Connor to give his presentation on the role of brain iron in neurovascular disease. About 6 years ago, I did a review on iron and read several hundred papers in the process, and I think most of them must have been his. And we really widely quoted and used a lot of his expertise in terms of measuring iron and understanding the role of iron, and it's a great privilege for us to have him here. Jim? All you really need to do is just hit this. You can just face the audience.
Okay.
And if you need a pointer and you want to turn, that guy will do the job.
Sounds good.
Okay.
Thank you. Mark, thank you very much, and I thank the organizers for the opportunity to be here. My only challenge in this particular talk today, I'm happy to share with you our work, but I found myself at each slide wanting to stop and ask the audience some questions. So I'll try to get through. So the neurological diseases that we've been able to map as being associated with iron mismanagement over the last 25 years or so we've been working in this field is Alzheimer's disease, and again, I know a lot of the imaging work trying to look at the iron burden of plaques that we had shown a number of years ago that iron's associated with neuritic plaques. New data just coming out of Ashley Bush's group, maybe about 5 years old now, that the amyloid precursor protein has functions as a peroxidase. Iron accumulation in Parkinson's disease, particularly in the substantia nigra, has been around for a while. And again, another recent paper coming out showing that knockout mice accumulate iron in the substantia nigra is being tried to tie that back into the peroxidase activity of the amyloid precursor protein. Basal ganglia seems particularly susceptible to iron accumulation with a number of the diseases that you see there. A lot of interest in this meeting in multiple sclerosis, and again, oligodendrocytes are really the most iron-rich cell, at least on a staining basis, that we see in the brain. And I mentioned restless leg syndrome here in a different color because all of the other disorders that I've mentioned seem to be associated with too much iron, at least what we're measuring in autopsy and on some of the MRI studies, whereas restless leg syndrome really seems to be an iron deficiency as far as the brain goes. And we've been spending time looking at brain iron transport, and I'll mention some of that. There was a question before, one of the earlier speakers this morning was in response to her talk, somebody was pointing out the number of movement disorders that seem to be associated with iron mismanagement. And if we look at the iron distribution in the brain, the striatum is iron rich, and in fact has equal to the amount of iron is equal to that that's found in the liver on a per weight basis. Pardon me. The substantia nigra, as he says, the most intensely stained area of the brain, deep cerebellar nuclei, red nucleus, and we tend to measure, although Mark pointed out some of this may need to be revisited with some of the new MRI and SWI data, but in general we tend to find more iron in white matter than in gray matter when myelin is normal, and that of course is consistent with the idea of the predominance of iron staining in the oligodendrocytes. But what one sees is, in general, these areas that we're talking about all have a functional relationship to movement. And here's just a picture that I like to show because it shows the consistency in brain iron status between human, where you can see basal ganglia. Went the wrong way, sorry. Can I go back? So you can see the basal ganglia here, head of the caudate, and putamen globus pallidus. This is a Prussian blue stain, so you can see the iron very intense in this hemisphere of the human brain. Over here you see red nucleus and substantia nigra also staining very intensely. Very similar pattern occurs in both mouse and rat brain as well, and particularly you see the enriched basal ganglia or striatum there. This is a slide that I've always loved but always questioned, and Mark and I were just discussing. This shows, as I mentioned, that restless leg syndrome we tend to attribute to a brain iron insufficiency, and here you see red nucleus and substantia nigra in a normal individual, and the absence of the hyperintensity in the RLS individual. This, I don't have a slide in here, but this really tends to be pars reticulata, not pars compacta. And this is where we see all the iron staining in the oligodendrocytes in this area, but it's also where the myelin is. And I always wondered if when we lose the hyperintensity in here, how much of that is iron and how much of that might actually be myelin, or perhaps even the microvasculature. Changes in that particular area. We have measured myelin in the RLS patients, both through MRI as well as autopsy, and found that there is a global hypomyelination. So I often wonder if this region might not be useful as more of a indicator of myelin status as opposed to only iron status, and perhaps this is something that could move forward out of this meeting. When we look at brain iron content, does it matter? Here we see memory function in hippocampus in a study that we did with George Bartzokas at UCLA, and here you can see the inverse correlation is significant for men. The higher their hippocampal iron status, the worse they did on a total word recall study, whereas in women the effect was not significant. So what we've really been spending our time on is how we regulate that brain iron. How do we account for the different brain regions? How do we the iron status of the different brain regions, and just in general, what is the flow of iron across the blood-brain barrier, and what's regulating it? So the sort of three most obvious, I think, places to start is it genetics, regional differences in the brain. There's certainly differences in metabolic demands, degree of myelination, synthesis of neurotransmitters in different areas, oxygen utilization is not uniform across the brain. And are there signaling mechanisms that might help us understand how these different brain regions regulate their iron? So if we look initially at genetics, a slide that I always found very interesting from Byron Jones at Penn State at our main campus at University Park. This is looking at a number of different BXD, these are inbred strains of mice, and I forget now the number, but 12, 15 strains of mice that he looked at, and as you can see, going from strain 13 up to strain 24, there's actually a threefold difference in iron content in the midbrain region of these mice. No dietary changes, nothing. This is purely genetic differences. One of the areas we've been spending a lot of time in lately is looking at the HFE gene, which is on chromosome 6, and it's an MHC class 1-like protein. Most of you in the audience probably know it as involved in hereditary hemochromatosis, but we've been looking at it in sort of the subclinical disease, if you will. People who carry the mutation still have higher levels of ferritin, higher levels of transferrin saturations, but the penetrance of the disease as far as having classic hemochromatosis is only about 1 in 30,000 individuals, even though if you're from Northern European descent, you have 1 in 200 to 1 in 400 odds of carrying the mutation, and if you're Irish, it's 1 in 100. The HFE gene is expressed in most tissues, including brain, and when we first started this was the big argument initially is that, well, the brain's protected in hemochromatosis, you're not going to get too much iron in the brain, why are you doing this? So we showed HFE in the blood vessels, so the argument became, well, you either have to accept the argument that when HFE protein is in the blood vessels, it doesn't work like it does anywhere else, and therefore the brain is protected, or the brain might not be protected, and we should go down that path. So it took a while to convince people to fund us to do this, but we eventually started to be able to move forward. So just a quick overview of what the HFE protein does. This is the transferrin receptor on a sort of a generic cell, if you will. If you have the wild-type HFE, it it binds to or becomes stoichiometrically related to the transferrin receptor, and it limits the amount of iron that enters the cell. So the way it reacts with the transferrin receptor or interacts with the transferrin receptor, only one transferrin can bind to the transferrin receptor, and only 2 irons then come in per binding event. The most common of the mutations is either the variance is the H63D, which still makes it to the membrane, but it doesn't stoichiometrically interact correctly with the receptor. So now for every binding event, 4 irons come in instead of 2. And the other mutation, the one that's more commonly associated with hemochromatosis, but rarer in the population, this HFE protein, mutant protein, does not make it to the cell surface, and as a result, again, excess iron enters into the cell. So we made a mouse model that carries, it says here, H67D, which is the mouse equivalent of the H63D mutation. So we knocked out the wild-type HFE and knocked back in the H67D mutation. So there's no wild-type HFE in these mice. And you can see within 6 months, there's elevated brain iron in the brains of these mice, both heterozygote and homozygote, and I won't say more importantly, but from a functional consequence, you see that the amount of protein carbonyls, which is a marker of oxidative damage to proteins, is elevated in these mice. We also did a study that we published, I don't know, about 8 years ago now, that looked at the frequency of the H63D allele in ALS, and since that time, studies coming out of China and Italy, the Netherlands, and the United Kingdom have all supported our findings. And what you see here is that there's a dose-dependent increase in risk for ALS if you carry the H63D mutation, that it ends up being about a 4-fold risk factor for ALS, which doesn't sound like a lot, but it's better than anything that's out there right now. As far as looking at HFE impact on brain function, here we see, looking at the working memory study, and this again was the study in the Barzokas paper, here that if there was no mutation present in in HFE or in transferrin, there was a correlation actually between loss of working memory and iron content to the basal ganglia. But interestingly, if one of the mutations or both were present, there actually no longer was a correlation between the two. It brings up one interesting idea in looking at the impact of these mutations on brain function, which is this recent paper that was published in PNAS that looked at H63D and found an increase. What they're reporting was an increase in white matter fiber integrity, and one of the interpretations here was that if loss of white matter is part of underlying or possibly underlying cognitive decline with age, perhaps an H63D mutation, which would allow potentially more iron and more myelin into the brain, might actually be protective in some cases. So again, as this audience certainly appreciates, iron tends not to come down on one side of a story or other. It can be good and it can be bad. So looking at the regional differences in the brain, I just wanted to share with you one study that came out of my colleague John Beard's lab many years ago now, and just kind of looking, the question was, does neonatal iron deficiency influence brain iron? And these are animals that were, iron deficiency was started at day 21 and then stopped, and the animals were carried out to adulthood. You have to sort of go over here to the black boxes. We start there for the control animals. And you can see that in some areas like the cortex, there was a loss of brain iron with iron deficiency. In the pons, there was no loss. Hippocampus took a big hit. Substantia nigra really didn't change from the control. But what happens in the substantia nigra is under iron repletion conditions, it actually overshoots the control level. And it's not still different from where it was for iron deficiency. But here's, again, a nice question of regulation and uptake with repletion. Here the hippocampus repleted back to normal, the cortex repleted back to normal, but only in the nigra did we see the overshoot. One of the things that might be very exciting with the imaging technology that we're seeing this week is looking at the dynamics of brain iron status. You know, we've been taught and learned that that brain iron is fairly static. That you get a certain amount in and it stays there, and we pretty much know that can't be true. There has to be a greater dynamism, and in this study, I think, which is kind of beginning to start to look at that, looking at total brain iron and looking at control versus iron deficient. So if you look at total brain iron in the controls, there was no different whether or not they were male or female, or the brains were analyzed after the animals were killed during the light or the dark cycle. But if you see here, if the iron deficient animals had lower brain iron, if the animals were killed during the light cycle as opposed to in the dark cycle, it didn't matter for the males between light and dark. It did for the control versus iron deficient. They did lose, but this difference wasn't observable if they went in if the animals were taken in the dark cycle. So it'd be, I think, fascinating to look within subject variation over light and dark cycle. I think there's just some really neat opportunities there. So how does the brain overshoot? How does the brain not change to iron deficiency? How does it overshoot with repletion? How does it just go back to normal? There must be signals, and that's where we are now, and kind of looking for these particular signals. So how is the regional heterogeneity not only established but maintained, and some of and if there is a signal, is it different among regions, and what are the cell types involved? So here is sort of the current model of brain iron uptake, and it shows the conduit model across the top. The transferrin binds to the transferrin receptor, and you transcytose this complex across the other side, and iron comes out once you get into the brain. A lovely paradigm that existed forever, basically, but you start asking questions. Well, why would it bind on this side and let go of the iron on this side? We know that this is a pH-dependent process, and the pH on both sides is pretty much the same. But even if you want to argue dissociation constants and things that might promote this, the other thing is this paradigm fails to address is the iron requirements of the endothelial cells themselves. We know there's a lot of mitochondria there. We know there's ferritin there. So somehow the endothelial itself must get access to this iron, and we've shown that there's DMT1, divalent metal transport protein, which is the symporter that allows movement of iron from the endosome into the cytosol. So we were able to show that is there. So we had another question then: is this iron only for the endothelial cell, or can that iron become available to the brain later on? So we set up this blood-brain barrier model, which enables us to, maybe I'll just use this for my pointer, it enables us to look at we put endothelial cells across here, we load them with iron, and then we put just regular media up here, and we can put whatever we want down here in the basal chamber and see if that influences movement of iron. So one of the things we did was we took CSF from monkeys, and these were monkeys who were either iron deficient or iron sufficient. And what you can see here is that in the CSF from animals that were iron deficient, much more iron was released from these endothelial cells compared to iron that was released from in response to iron sufficient CSF. So there's clearly a signal in the CSF that is informing the brain endothelial cells about how much iron to release. and later we're going to have talks about hepcidin and ferroportin, and we think both of those are actually involved in this particular model. But the other thing we were excited to show is that there was a significant correlation between blood parameters, so the amount of iron that was released correlated positively with both the mean corpuscular volume and the hemoglobin contents of the monkeys. So in summary, as I said, I put the summary together before. I just wanted to ask you guys a bunch of questions. But certainly what we know is that the current paradigm for how iron is interacting with the neurovasculature to be inadequate, and how much of that could, with some of the novel technology, could that be evaluated in the living patients with the tools that you have. We know that endothelial cells require iron. We're now arguing or postulating that the endothelial cells are actually a reservoir for brain iron and not simply a conduit. And furthermore, we're arguing that the site of regulation for brain iron uptake is actually at the level of the endothelial cells, and certainly that the brain iron content is dynamic, and the diurnal variation data tend to support that. So your take-home message here is that iron transport into the brain we know is affected by gender, age, genes, and diet. And I know MRIs are expensive, and you don't want to have to evaluate all of these differences. But so as a nod to that, I left off the diurnal variation part. But I think the within subject changes would be exciting to evaluate as well. And what we're looking for is how this process now is regulated. And as I said, we've shown now some evidence that there's proteins in the CSF that and CSF is in dynamic equilibrium with the extracellular fluid. So what's in the CSF is going to probably also be in the extracellular space, and so we're looking for the source of those proteins that are helping us regulate the iron. So. Just to acknowledge Padma Panuru, who's done a tremendous amount of the blood-brain barrier work, as some of my other colleagues that have been involved in some of the transport studies, students in the past, Joe Berto and Tom Dickinson especially. So thank you.
Jim, thank you very much. We will, I'm sure, have some questions for you. I would just like to comment. You may have answered a question that we studied about 5 years ago, comparing the India ink injected cadaver brains by Henri Duvernay, and in his 1999 book on the vasculature of the midbrain, he shows that there are variations in the red nucleus and in the substantia nigra that he doesn't correlate directly with iron at all. And I'm wondering if this iron in the endothelium you're talking about might be more at the capillary level, because about 5 years ago, this paper we published showed that with SWI, you could see the variations in the pattern in both those areas in the midbrain that exactly matched Duvernay's images. And we checked by using contrast agent, we checked bold, and in fact, none of those changes can explain the fact that we see this high iron content in exactly the same pattern. And I'm wondering if then the endothelium just at the capillary level could have more iron in it than the bigger vessels, for example.
No, I think that's probably where the regulation is taking place.
David.
Even before CCSVI came along, we've known in the lower extremities that you have iron hyperpigmentation with varicose veins and with and iron deposition. And it seems as if everyone just assumes that this is microhemorrhage. But it always seemed to me possible that there was a problem of overloading the endothelial transport system. Have these issues been settled?
No, not at all. And I think you're exactly right in the way you're thinking that we should be looking at this, because it's always either there was the conduit or a microhemorrhage, and there wasn't something in between. But I think the data now are pretty clearly, certainly all the transport, all the storage proteins, and the release for iron inside from inside the cell are all found within the endothelial cells. And we already know they have mitochondria, so they have to have some source of iron. So I think as we move forward with this, asking those specific questions, how much iron are they willing to store? Again, what's the diurnal variation? Is there a problem in storage with disorders like restless leg syndrome and the perivascular diseases as well? And I think it goes in farther, because one of the you always look at these brain iron uptake studies, and they say, well, we injected iron into the brain and we measured what came out, and there's 50% less came out, so 50%'s in the brain. It might be in the microvasculature. It's not necessarily in the brain parenchyma, and I think those are not just academic esoteric questions. I think they go to brain function in disease states.
Randy, just one short question, please.
Sure. Yeah, it's just really more of a clarification question. You pointed out the heterogeneity in the distribution of iron across the brain. I think what you were saying was that motor structures seem to have, in general, a higher level of iron content, right?
Right.
And is that related to transport chain? Is that metabolically related, or we don't know?
We don't know. We don't know. In the basal ganglia, you could argue metabolic related, but no, I think those are the questions that we're just now getting the tools to answer it. And metabolic, maybe myelin, maybe vasculature. I think there's two other options in addition to metabolic, although they probably all tie in together.
Thank you. And Bill, last question.
Well, just with regard to being that oligodendrocytes are really high in iron, and so is early formation of white matter, inhibited when there's microcytic anemia and so on, but is that individual more vulnerable later for oligodendrocyte breakdown, or is this oligodendrocyte breakdown another potential source in demyelinating diseases of this excess iron in the brain that we're seeing?
Yeah, no, I think it could be another source. At some point, it increases the vulnerability of the oligodendrocytes to pro-inflammatory cytokines. Subsequent to that, they're going to release their iron and just exacerbate the situation. I just finished writing a review, and one of the questions that I asked in that is that we always see iron-positive oligodendrocytes in patches, and I don't know if the resolution is there or not yet, but I thought one of the really interesting questions would be to see in terms of MS lesions, do they overlap with these patches, or are there why are there areas of vulnerability in MS, and could we map those to the patches of the iron-positive or iron-enriched oligodendrocytes?
Thank you very much. Thank you, Jim.
Thank you.
Does this work? So the next presentation will be given by Donato Ghimati on hepcidin and ferroportin genetic variants in MS, susceptibility and progression.
My talk comments about the possible relationships that may exist between the common iron polymorphism in the main important iron genes and the main clinical characteristics that can be measured in multiple sclerosis patients. We know that brain iron accumulation is a common finding in MS and also in other neurodegenerative diseases. We know also that as iron increases in the brain, the disability of patients, not only physics, increase. Regardless, CCSY, yes or not, whatever the mechanism causing brain iron deposition is, the same group of proteins, the same group of cells, the same group of genes work the iron balancing, and it is finally tuned by influx, efflux, and storage steps. So we looked at the common SNPs. SNPs means single nucleotide polymorphism within the main iron protein genes. These different steps involve different receptors, and among these, the ferroportin epsilon axis dominates the iron balancing. That is critical for iron homeostasis. This slide shows you a particular cell that is the macrophage. This is important in several degenerative diseases in which inflammation and autoimmunity may be present. Just to explain you how the ferroportin epsilon works in few words, we know that when iron is high in the blood, the liver synthesizes epsilon, and the epsilon move toward its target, that is the ferroportin, and destroy it, because we do not need that iron increase in the blood from the storage that we have in the cell. Few information about how basically several iron gene works. You should consider the gene not like a piece of DNA that are statics. They crosstalk with the environment, with the cell, and also with each other. In iron deficiency condition, the left panel of this slide, we have that when iron is low in the cell, we need to increase the recruitment of iron, so we need receptors. In the end, 3' end of the gene, that is this one, we have receptors for iron regulatory proteins that are free to bound this region because we have no iron in the cell. What's happened? That the mRNA of the gene is more stable. So several cycles of synthesis of the protein happen. On the other hand, in the same iron deficiency condition, another gene, the ferroportin, that has a different task if compared to the previous one, carry this switch. At the beginning of the gene, this is the promoter of the gene. When the iron regulatory proteins bind the iron regulatory elements at the beginning of the gene, we suppress the synthesis because we no need that the few iron that we have in the cell go out. What's happened in iron overload condition in the cell? The same gene can decide if help or not. This time we have a lot of iron in the cell, so the iron regulatory protein prefer to bind the iron regulatory protein and the iron. So we do not have a lot iron regulatory protein that can bind this region of the gene. So we will not have synthesis because the mRNA is less stable than before. Of course, now it is easy to understand that the ferroportin now, with a cell that is full of iron, can open the gates, so the iron go out the cell. And now, just to close the circle, when the iron increases in the blood, the liver will synthesize epsilon, and the epsilon can feed back in a negatively way the ferroportin. Now I introduce you the two different genes that I have investigated. Within the promoter of the ferroportin and epsilon, there exists two gene variations. These gene variations are not so rare, and we know that about 55% of general population carry this variant. As right of this kind of polymorphism, till now we do not have phenotypic described effects. But we think that due to this position, -8 CG substitution, very close to the iron regulatory elements that I described you before, we think that could have effects on transcription. Because don't forget that within this region we have the switch to decide to off or not the protein. According to the similar description for this gene, the epsilon contained in the promoter of the gene another variation named -5A2 AG substitution. Conversely to the previous one, we know, even if this is very recent to describe, that the presence of the polymorphism, I mean the G allele, affect by about 20% the synthesis of the protein. 20% is enough. You think that in a chronic disease in which iron can increase by a lot of time, we affect partially the answer that epsilon could have against the ferroportin. And this is just to show you how the switch can be pushed on or off. This action destroys the synthesis of the gene. Well, the aim of our study is to analyze whether or not these two common polymers I described you contribute to the main clinical characteristics of the MS patients according to disability, severity, progression of the patients. We perform a case control study and also a intra-case analysis. This slide shows you how many subjects we analyzed, split 50 and 50% cases with MS, and completely matched healthy controls. In the last line, you can see how many patients belong to the different clinical course of the multiple sclerosis. Don't worry for a lot of number of these slides. We can comment just a few significant findings. Accordingly to the disability, progress index, and severity, of course, the progressive subgroups show higher mean value if compared with the whole group or with the relapse-remittent subgroup. The age and the ratio of male-female was comparable. Major findings of my investigation. Look please to the red line. We looked at the homozygous condition of the ferroportin gene, and we found that among the whole group of patients, we found an overrepresentation. 20 people carried the homozygous condition, and they account for 7% of cases, if compared with healthy controls in which the percentage was less, 1.7. This overrepresentation was strongly significant, and this was responsible for a risk associated to multiple sclerosis of 4.4. Sub-analysis did not show a large difference between this kind of overrepresentation among the different clinical phenotype. Okay, it's that, maybe. We note just that among the secondary progressive subgroup, 5.6, yes, you can see, risk was obtained. No significantly higher with respect to the other one, but just to note. So we can say that the homozygous condition of the ferroportin polymorphin associated with a risk for MS of about 5-fold. When we looked at the other gene, the epsilon, we obtained completely different results. The first thing you can note in this slide is that among relapse-remittent subgroup, we did not have overrepresentation of the risk allele. And this number is completely matched with the healthy control percentage. You can see that among progressive, analyzed together or separately, the percentage, the rate of homozygosity was statistically higher. At the end of my speech, I'll try to discuss with you and to explain why this happened. For the moment, we can say that homozygous condition of the epsilon gene may associate with severe MS phenotypes. But we could ask, could be this a prognostic marker? Could be this a marker that tell us that who carry this kind of gene, risk gene, may become rapidly or may have a more severe clinical phenotype? I'll try to answer at the end of the speech. Now we look at the severity score of the whole group of patients and stratify the mean values according to the 3 different genotypes of the ferroportin gene. We obtained that the severity increased as the number of the polymorphic allele increased in genotype of patients. 0 allele, 1 allele, I mean the G1, and 2G allele. So we state that the severity of the whole group of patients increased according to the number of the polymorphic risk allele. Same analysis for the epsilon polymorphism. The same situations happened, but the significance was higher. And the mean value of the severity score of the homozygous people carrying this was very high. Same conclusion. Now, just to answer to the question that I raised a few minutes ago, and could be the hepcidin a prognostic marker that tells us something about the future, the fate of the affected patients. We try to answer and to measure this. We try to measure how long was the time in which our SP patient had remained were in the previous less severe clinical phenotype before to become secondary progressive. We performed a retrospectively analysis, and we had a follow-up of 10 years and analyzed all our secondary progressive patients, 103, and we split them in two different groups, those carrying and those not carrying the risk allele. The first thing that you can see is that among the patients that carry the risk allele, starting from the first year after the diagnosis of MS, when they were relapse remittent, patients start to become progress, and years by years, the number of progression increased. Not only if we measure the global number of patients, but also the rate, the velocity of patients was higher. When we compare with the patients that did not carry the risk allele, we can note a different behavior, but the important thing is that among this subgroup, no risk allele, nothing happened before the fourth year. Maybe a chance this because we have few patients. Anyway, the two subgroups are balanced. Just to reinforce this observation, we included in the same analysis also the relapse remittent patients. The relapse remittent patients are till now relapse remittent. They did not progress. So the same analysis now show you that for the progressive patients, the endpoint of this Kaplan-Meier analysis is like before, the exact date in which they became progressive. For the relapse remittent, the endpoint is just the duration time starting from the diagnosis. If the gene allele does not influence the disease duration, this significance could disappear, but we obtain that increase. And the last proof could be this. If we measure merely the mean value of duration time started from the beginning of the diagnosis, all our relapse remittent cases, and stratify according to the genotype, we obtained that those carrying the homozygous condition, the left green dots, show the shorter mean duration time. And the duration time increase according to the disappearing of the risk allele. Now can state that the absent allele may be considered something that associates to an early progression. If I am a relapse remittent patient and I became MS patient today and I carry a risk allele, I could have a higher chance to progress earlier if compared with relapse remittent patients that did not carry the risk allele. Now, do you remember the slide that I showed you a few minutes ago? We now could try to explain why, according to this kind of polymorphism, no or few homozygous people belong to this less severe clinical phenotype. We could speculate, we could hypothesize that the patient that start to become relapse remittent had, if they have a higher chance to progress, this split, this switch, this became progressive. So this is responsible for two different findings. One, among the secondary progressive, the frequency of homozygous people increased because the homozygous people that are relapse remittent switch in this second clinical phenotype. And, of course, among relapse remittent patients, the percentage of homozygous decreased. Last slide. Just a moment, sorry. Since these two polymers are common among general population, one patient and one people could carry both risk allele. So we did a combined analysis and tried to find how many people, how many patients carry the double risk factor. We found that the MS patient, the red block, carry 12% of double carriers. If compared with healthy controls, the green blocks that show you 5%. On the counterpart analysis, we've measured also how many people do not carry any risk allele. And of course, now the ratio is inverted. Among patients we have low normal allele, and among healthy control, the percentage 18% increase of people that carry zero risk allele. Analyzing this statistically, we have the risk for MS of 3.5. And finally, findings, when we compare these two subgroups of patients, according to the disability, the progress index, and the severity, we obtained that all these parameters were in the mean value higher, significantly higher, than the counterpart of patients that were fully wild-typed. I mean, zero risk allele. In the last line, you can see the strong significance of this kind of comparison. Conclusion. Iron misbalancing is a key point in MS, and the wide clinical variation that we observe in such a complex disease may be in part genetically predetermined. To look at common variation may be strongly helpful in clinical diagnosis prognosis, and to have in our hand a prognostic tool markers could help strongly clinicians and patients. I want to thank all the people that worked to this investigation. Thank you very much.
Thank you very much for this quite interesting presentation. The presentation is open for question. Actually, I have one.
Yes.
Is there any idea how the results from the genetic analysis could relate to iron accumulation in the brain tissue?
Yes. Different to the previous speech, we did not say exactly how many iron could be in the brain or in the central nervous system. We just jumped these steps and we assessed the frequency, the genotype distribution in patients, and important, I think, also the clinical findings, because this is quite translational medicine. Of course, if the gene has a role is that iron homeostasis is affected, because this kind of polymorphism may and are sites on the crucial DNA region of this, and a lot of paper are present not for these two very recently described polymorphism, but for HFE, the two main polymorphisms. There exists correlation between iron accumulation in tissue, not only brain. So we think that the next step could be this, to include in the steps the second one. That's very important. Thank you.
It would be very interesting.
Yes, yes, this should be. We should also increase the number of patients.
Yes, please.
The question is, how is it with venous ulcers? Do they also show comparable developments? Because there is a similar mechanism involved, according to my thinking.
Yes, as you know, I started with Professor Zaboni to work on iron gene in this kind of chronic venous disease, degenerative disease. And I noted this, that the value of risk, that is the statistical tool that can help us to understand or not if it's something that is hard to say, like a gene, like a variation, and the clinical phenotype. We found that also the number that the outputs that we obtained are completely the same. 6, 7, I remember for HFE, that is another gene, but the mechanism is the same. We obtained 6, 7-fold increased risk when the two conditions coexist. I want to say this last sentence. This kind of polymorphy do not carry and are not responsible for the disease. But when two different situations that may be other affected gene, but also the environment, maybe the iron accumulation due to venous stasis. When two different things coexist in the same patient, we can have the severe clinical phenotype, like for venous leg ulcer.
When you have a homozygote patient, with this challenge, is there value to very meticulous attention to their hemoglobin by treatment of...
To the hemoglobin, sorry? What?
Well, by lowering their hemoglobin in a consistent manner, much as you would a hemochromatosis patient, bleeding them.
No, I do not look this, but I think not, because these are not mutation. If you have a mutation in the gene, you could have systemic iron overload or anemia if the problem is in the other side. But this substitution, this very frequent mutation, are responsible of the disease just in a localized.
Okay, thank you.
Yes, this can have effect in the legs if we have stasis in the leg, or in the brain if you have overloading of iron in the brain. So I think not.
I appreciate it.
Thank you.
Thank you. We'll have to move on. Thank you very much. Our next speaker is Stefan Ropola, who I had great pleasure in meeting in Graz a few years ago when we were discussing iron and MS. And he has published quite a bit in the last few years and is an MR expert and is going to share today his experience in not just measuring iron with T2 star and other methods, but as it applies specifically to multiple sclerosis. Stefan, thank you.
Thank you very much, Mark. Okay. Thank you very much for this kind introduction. And I also would like to thank the organizers for inviting me. So I have nothing to declare. I think it's commonly accepted that the iron metabolism is significantly distorted in MS. However, the causes as well as the consequences remain largely unclear. To learn more about iron deposition, we need sensitive and also non-invasive technologies. And in this context, MRI appears as an interesting candidate because it is non-invasive and it allows it is sensitive also for the paramagnetic effect of iron, and it's readily available. So in my talk, I would like to summarize what we have learned so far by applying MRI to assess iron levels in MS patients, but also what are the limitations and what we can expect from the future. So actually, first observation that the iron level is increased in MS patients were made with histology, not with MRI. The picture we have from histology is rather limited for many reasons. First of all, only a limited number of studies have been performed so far with a limited number of samples. And typically these samples were taken from regions with macroscopic lesions and also from patients that usually died from MS. So the pathological condition of these samples may also differ from what we can see in younger patients or patients with a less aggressive course of the disease. So what has been observed so far is that iron accumulate, and what I also would like to mention is that most of these studies have been done with pearl staining, but it has turned out that pearl staining may miss or can miss a lot of iron in white matter, in particular in lesions. So DEB-enhanced staining is more appropriate. But what we have learned so far from this observation is that iron accumulates in lesions and in around lesions, mostly at the rim of lesions, in vessel walls and also around veins. And the conclusion that have been drawn so far or explanations for these findings are disruption of the blood-brain barrier, reduced external clearing, but also attraction of iron-rich macrophages and maybe also some chronic inflammatory damage to the vein walls. So it seems that iron accumulation may reflect a multiplicity of pathological and physiological processes in MS. The first observation with MRI date back to the very beginning of clinical MRI, which was at the end of the 80s of the last century. Dreyer and colleagues noticed hypointensities in the basal ganglia on T2-weighted spin echo sequences, and they subsequently performed a histological correlation study with pearl staining, which colors all non-heme iron blue. And they could nicely demonstrate that the T2 shortening was because of the presence of iron. So iron is paramagnetic and produces microscopic field gradients, and when the spins or the water protons diffuse or tumble through these gradient fields, they accumulate an arbitrary phase shift which cannot be fully refocused in a spin echo sequence. They consequently also looked at the signal intensities or the basal ganglia in MS patients, and with simple visual rating score, they could nicely demonstrate that MS patients with more severe MS, they had more pronounced hypointensities. So they concluded that the striatal signal intensity could be used as a marker for disease activity. It's commonly assumed that the iron in the basal ganglia is ferritin. Ferritin is a global storage protein which keeps the iron in a non-toxic and soluble form. And each of these ferritin shell here can host up to 4,500 ferric ions, which form a crystallite, very similar to the mineral ferrihydrite. Overall, in the total body, ferritin makes up only up to 25%. However, in brain tissue, it's the most frequently found iron component. In the last few years, a couple of MR methods have been proposed and developed for iron mapping in the brain tissue, including relaxation time mapping, magnetic field correlation imaging, quantitative phase imaging, also known as assessment of the local phase shift, direct saturation imaging, and also susceptibility mapping. Apart from DSI and susceptibility mapping, all these methods have already been used to assess iron levels in the MS brain. And I should also point out that they have been used exclusively to assess iron levels in the basal ganglia. The strongest predictor for iron accumulation in the basal ganglia is age. After birth, there is no ferritin in the brain, but there is a progressive accumulation of ferritin as a consequence of normal aging in different structures of the brain. So most of our knowledge on iron accumulation as a consequence of normal aging comes from the work of Holgreen and Zarenda, which dates back to 1958, and which is still the largest postmortem study with chemical analysis of brain iron. In MS, there is a lot of evidence that there is additional iron deposition in the basal ganglia, which is linked to disease duration, to disability, to cognitive decline, and atrophy. And interestingly, the strongest disease-related effect typically can be observed in the putamen. Also, the highest iron concentration can typically be observed in the globus pallidus. So this suggests that iron accumulation in the basal ganglia therefore suggests that this reflects accumulated neurodegeneration. So let me briefly come back to the MR methods for iron mapping. The question is, which approach is the best in terms of sensitivity and robustness and clinical applicability? And this is not easy to answer, but what I can say is that mapping of the R2 star relaxation rate is an interesting candidate because it's just based on a simple spoiled gradient echo sequence with at least 2 echoes, but such a sequence is readily available on the most clinical scanners. It can also be performed in the 3D acquisition mode, which provides high SNR and also high resolution. And I should mention that R2 star can be calculated by fitting a single exponential decay function in the signal intensity from the individual echoes, and also by considering noise, in particular for later echoes. Another argument for R2 star mapping is that it's so far the only fully validated method. So we recently have performed such a validation study where we have done post-mortem MRI. It's retesla in situ, which means we had measured R2 and R2 star in the brain before the brain was extracted. And then after MRI, samples were taken from different predefined regions in gray and white matter and were referred to mass spectrometry, which allows detection of elements at a parts per trillion level. And what we found were that both R2 and R2 star nicely correlate and show a strong linear correlation with iron concentration over the whole physiological range. However, the strongest correlation was found for R2 star. So, interestingly, maybe they cannot be seen here that clearly, but this correlation was only very weak for the white matter. The white matter is represented here by these blue crosses. We recently did R2 star mapping in a larger cohort of MS patients, and one of the most important outcomes was that the strongest predictor for iron accumulation in the basal ganglia is cortical atrophy, but also atrophy of the deep gray matter structures. And in this study, we also had included a larger cohort of patients suffering from clinically isolated syndrome, suggestive of MS, and we were not able with this method to probe increased iron values in the basal ganglia. So the fact that iron levels seem to be not increased or not that much increased at a very early stage of the disease suggests that this is rather, the iron accumulation in the basal ganglia is rather an epiphenomenon of MS and just reflects accumulated neurodegeneration rather than a trigger or a promoter of the disease. So, but that could be totally different in white matter because iron in white matter could be more directly related to the disease activity. We know that iron produces oxidative stress and free radicals, which in turn can trigger or at least promote the inflammatory and neurodegenerative cascade. So for these reasons, we and also some others have recently focused on R2 star mapping in MS lesions. And what can be typically be found is that R2 star in most lesions is reduced. So this can also be seen here on these images. If you look at these periventricular lesions, in the corresponding R2 star map, these regions appear hypointense, meaning R2 star is lower. Does that mean that there is less iron in the lesions? I would say no, but we have to be careful because what comes into play is the diamagnetic effect of the myelin. The myelin is diamagnetic and also produces microscopic field gradients, which also increases R2 star. So another explanation for this observation is that it's just demyelination. But on the other hand, we can learn that R2 star mapping is not a very reliable tool to assess iron levels in the brain. In this context, much hope is put on susceptibility mapping because the magnetic susceptibility is directly related to the iron content. The magnetic susceptibility is a physical property of a tissue that tells us how much it becomes magnetized when we put it in a magnet or in a scanner. And the resulting local field shift here is then simply a function of the unit dipole function. It's a convolution of the unit dipole function with the bulk susceptibility, which corresponds to a multiplication in the Fourier domain. So actually, we can measure the local field shift with phase images coming from a gradient echo sequence, but actually we want to know chi. So mathematically, this is known as an inverse problem, but this is not easy to solve because of its ill-posed nature. So just in the last year, a couple of new proposals have been made how to solve this inverse problem. And one of the most promising approaches are the morphology-enabled dipole inversion, which incorporates also the magnitude images of the gradient echo sequence, and the sharp approach, which is based on the assumption that the source of the background phase is located outside the region of interest. Oops, wrong, sorry. So the local field shift can be easily assessed from the phase images, but first we have to get, sorry for that, we have to get rid of these phase wrapping artifacts, which just represents turnovers from 360 degree to 0 degree and vice versa. And we also have to get rid of these unwished background phase. So typically image processing steps incorporate homodyne filtering and high-pass filtering, so that we will end up with this corrected phase image that is directly related to the local phase shift. Some have proposed to use these local phase shifts or field shifts to assess iron levels or to estimate iron concentration, but there is increasing evidence that this is not a very reliable approach. First of all, the local field shift also reflects some global susceptibility effect. And another issue is that the phase value is changed due to the filtering that is applied to get rid of the background phase. So this is data from Bing Yao and colleagues showing that the phase value strongly depends on the magnetic field strength. It makes sense. But overall, there is no clear correlation between iron concentration and local field shift. And this is also in line with our post-mortem data. This is still unpublished data, but showing that there is no clear correlation between iron concentration and local phase shift. So in that case, we have measured iron levels with ICP mass spectrometry. So phase images also form the basis for SWI, which is a very interesting and also intriguing new approach, and which is based on the combined analysis or combination of the magnitude and phase image. Mark Hakey has written an entire book on that topic. You should read that. Unfortunately, SWI is not quantitative in the sense that it provides values that are reproducible or can be compared among subjects. However, it provides a new and interesting image contrast that seems to be complementary to conventional MRI, because we can see lesions that are not visible on conventional MRI and vice versa. And in particular, as seen here, the rim of the lesions can be depicted much clearer than on conventional MRI. And it's also very well able to depict veins, in particular smaller veins that are draining lesions. But SWI cannot distinguish in these veins the effect from, or cannot distinguish deoxygenated blood from iron in the vessel walls, because both have the same paramagnetic effect. What you can see here is a quantitative, absolute quantitative susceptibility map, which we could reconstruct by solving the inverse problem with the MADI approach. Actually, this is not an MS patient. This is a patient suffering from small vessel disease. But I'm just showing this case to highlight the potential of QSM, because it allows to depict the different magnetic properties of iron and calcium. So iron is paramagnetic, therefore these microbleeds here appear dark, and calcium is diamagnetic, therefore this calcification in the plexus corrideus appear bright. So in contrast, the magnitude of this T2-weighted gradient echo sequence is just sensitive to microscopic field gradients, irrespective if they are caused by positive or negative magnetic susceptibility. Therefore, both conditions appear in the same way. Here you can see a QSM map, reconstruction from an MS patient. And now I'm sorry for that, now the contrast is inverted. I hope that doesn't confuse you. So myelin is diamagnetic, therefore it appears dark, and the cortex is more paramagnetic because there is less myelin and more iron in there. Therefore it appears bright. So we recently have performed a post-mortem validation study because we are interested how reliable and how sensitive is QSM for iron mapping, in particular in the white matter. And again, we found a quite nice correlation over the whole physiological range of iron concentration. But again, we found that this correlation was much weaker in the white matter. So what again comes into play is the diamagnetism of myelin, which counteracts the paramagnetism of iron. And interestingly, if you look at these lesions here, they are bright, which means they are paramagnetic. But another explanation could be also there is less diamagnetic myelin. So that's obviously a strong limitation of these susceptibility-related methods. Nevertheless, it seems that QSM provides a new image contrast that could also be of interest for MS. If you look at these cortical bands here, which are facing these lesions, they appear to be strongly paramagnetic, which is not visible on the FLAIR image. So maybe this is a new approach to depict cortical lesions. And one thing I would also like to mention is the slope of this correlation function, because the slope is in very good agreement with the slope predicted from theoretical consideration for ferritin. I should mention that the magnetic property of iron also depends on the spin state, and the spin state may vary between different iron compartments. So this is another proof or another evidence that most of the iron in the brain is ferritin. So it's obvious that a current strong limitation of these approaches is that the diamagnetism of myelin is counteracting the paramagnetism of iron. And in addition, iron concentration in white matter is usually rather low, so that further augments this mechanism or this effect. And I should also mention that in the meantime, there is increasing evidence that the susceptibility of myelin strongly depends on the orientation of the myelinated fiber with respect to the main magnetic field. So this is a further limitation. So let me briefly summarize what we have learned so far from applying MR methods for assessing iron levels in the MS brain. There's, I think, a lot of evidence that iron accumulation in the basal ganglia is mostly or more likely an epiphenomenon of the disease, and it mostly reflects accumulated neurodegeneration at its scales with increasing brain volume. And there is also evidence that most of the iron in the basal ganglia is ferritin. And the take-home message is actually that we are able to precisely measure iron levels in the gray matter and in particular in the basal ganglia, but not in the white matter because of the confounding effects of myelin and iron. So in this context, much hope is put on the application of these methods at higher field strengths, because that increases. I didn't mention that the susceptibility effect increases with field strength, so this provides a higher sensitivity. But we also need new approaches for mapping the myelin content so that we can disentangle effects from myelin and iron. So and with this last slide, I would like to acknowledge my co-workers who are working on the iron mapping stuff, and also our collaborators. Thank you.
We have a question, David.
Accumulated neurodegeneration, where's it coming from? Is it dead oligodendrocytes? Is it microhemorrhage? Why is it accumulating in the basal ganglia?
I mean, atrophy stands at the very end of the neurodegenerative cascade. We don't know the individual steps, but tissue that has gone is a consequence of atrophy. So that's what we define as accumulated neurodegeneration.
Why would
but we have no
iron?
idea what's going on exactly.
So, I mean, atrophy, why would atrophy have iron in it? Dead macrophages, perhaps?
So we don't know. I mean, this is a different process. This is a consequence, but we have no idea. I think nobody has an idea how the iron comes, for instance, from the cortex to the basal ganglia. I can't answer that.
Sorry, I think we'd better go on here. Can you talk to him perhaps during the lunch break?
I thought you had an answer, Mark. I think you're ducking it.
All right, Stefan, if you can introduce the next speaker. Thank you very much, Stefan.
So the next speaker is Kabil Habib, and he will present his work on assessing abnormal iron content in deep gray matter of patients with multiple sclerosis versus healthy subjects.
Thank you, Dr. Rapello. Well, it's a great pleasure to be presenting my work at the second ISNVD meeting. I would like to thank the collaborators who have worked with me on this project. Iron is a very basic element that is needed for brain metabolism and is important for oxygen transport and many functional effect in the brain. So when it behaves badly, as Cal said in his review, this will lead to unfavorable outcomes. In multiple sclerosis, we see that iron appear in the MS lesions. We see that it appears in the basal ganglia, thalamus, and midbrain. Also, we see that it shows an integrate pattern along the venous drainage over time, suggesting a probable correlation with venous abnormality. In our lab, we've seen using susceptibility-weighted imaging, iron deposition in the basal ganglia. So as we see that the caudate nucleus and, like as we see in the red arrows, the caudate nucleus and the globus pallidus has shown iron deposition. And if we look at figure B, which has been taken for the same patient after 2 years, we see that iron does increase. Now the critical question is this iron deposition normal? And is this increased due to MS, or is it just normal aging? So to answer this, we need to compare MS patients to normal. So the study we did, we studied 52 MS patients, 31 relapsing remitting and 21 secondary progressive. The mean age was 43. The mean disease duration was 8 for relapsing remitting and 19 for secondary progressive. And the mean EDSS score was 2.19 on average for relapsing remitting and 5.92 for secondary progressive. So we used susceptibility-weighted imaging. We used the phase information in order to measure the phase shift and as a mean to assess iron content. The structures we studied were the head of the caudate nucleus, the globus pallidus, putamen thalamus, pulvinar thalamus, red nucleus, and substantia nigra. Previously, we studied 100 normal controls, and we created a baseline of normal iron increase with age. So we used this as a baseline for this study. What we did, based on what we got from the 100 normal, we created a two-region analysis concept to study iron increase. What this helps is that if you have a large structure, if you have a subtle iron increase, if you average out, it's going to be showing no change. But if we actually separate it into two regions, even the subtle change will be able to measure it. And the parameter we used were average iron content in this high iron content that we defined it as being 2 standard deviation higher than the normal. We measured the total iron content and the normalized area. Now, this might look like a complicated table and a lot of numbers, but I'll make it as simple as a take-home message. This table actually represents the percentage of MS patients and healthy control who have 2 standard deviation higher than normal in each structure and 3 standard deviation. So if we look at the left side, we have subtotal for normalized area, average phase, and total phase, which were our criteria, and they all are embedded in the total. So briefly, I'll just show what we got in the total. And this says that basically if the patient have high iron content higher than 2 standard deviation in at least 1 structure, using any parameter, he will be considered for this. So what we saw, that only 13% of population between 20 and 40 years old had high iron content. However, when we consider MS patient, it was 78 for relapsing remitting and 75 for secondary progressive. And 41 and 70, as we see, we also see a difference between normal and MS patient. What was really striking is that when we consider 3 standard deviation, and if we take a look at the age between 20 and 40, only 1% of the normal controls had high iron content in at least 1 region. Well, 50% for secondary progressive and 67% for relapsing remitting. And combining all ages, we saw that 13% had high iron content, in normal controls compared to MS, where we see 67 and 65%, and this is statistically significant. So what can we conclude out of this? Iron increase are really good visualized using this concept. So if we have a localized high increase in iron content, we can see it using this method. We saw that the caudate nucleus, pulvinar thalamus, red nucleus, and substantia nigra has shown to be the most susceptible compared to the rest of the structures. We were able to see, too, that red nucleus is dominant. So we see high iron content in the red nucleus in older patients, and when I say older is when we divide it into 20 to 40 and 40 to 70. So we saw it more dominant in older patients, where we saw the pulvinar thalamus that shows 27.9% of the MS patient has shown high iron content, and they were dominated by young adults. So we believe that iron, the pulvinar thalamus, and substantia nigra might present a potential biomarker for MS. Thank you.
Thank you very much. This paper is open for discussion. I'm actually wondering how you build up your reference data or your reference phase. Was that by linear regression or?
Yeah, it was a linear regression.
And was this done for the over the whole age range? Or
Yes.
Okay.
Yeah. So we studied, the population was between 20 and 60 years old. It has been published in JMRI last year. And we did a linear regression, and we took the mean plus 2 standard deviation to be considered as high iron content.
Because from the whole green work, we know that there is, or we could expect a plateauing effect in the after the year of 30 or 40. So that would more speak against the linear regression.
Okay.
Yeah.
Stefan, just to answer that question, that he correlates this directly with the normal growth of iron as we rust, as we get older. That's already accounted for in this. So that's subtracted out, and then it's what's remaining after the normal age increases in iron are taken into account.
Okay, thanks.
Thank you. So we'll go on to the last presentation for today, iron deposition and disability in MS, and Dr. Zavadinov is going to give that.
Choose.
Oh, I think actually the presentation by Jesper Hegemeyer has not been received yet, so if that is not received, we will end with your presentation.
But I will present that one.
Oh, you are giving it. All right. Thank you very much, Robert.
Okay. So I will be short. First of all, thanks to the committee for choosing these abstracts. I have to say that unfortunately Jesper Hagemeyer could not come here today, but he did all this work and he's doing a lot of iron studies in our center. He's a fellow, a very bright fellow from Amsterdam who already works for a year and a half in our center. This study is in press now in American Journal of Neuroradiology, and these are our disclosures. In terms of the study population, there were 65 healthy controls and 42 CIS patients. The mouse works. As you can see, the age was slightly higher, about 2 years in healthy controls than in CIS patients, which clearly would be against our hypothesis. I have to mention and remind also Dr. Ropele that in their recent published study in neurology, there was approximately over 3 years of higher age in the healthy control group than in the clinically isolated syndrome, which could contribute to some of the findings that you reported in the recent neurology paper. Now, you can see that disease duration was 1 year, so very early, and all these people had only one clinical attack, and you can see the distribution was most of the patients have been under 9 hyperintense lesions. Just to point out very importantly, no differences have been seen in the total gray matter volume, white matter volume, whole brain volume, or lateral ventriculars between the healthy controls and CIS patients. So I would like to add to the discussion today, and I really think when you compare high-pass filtering on SWI, it really matters, as Dr. Ropele pointed out, which type of filter you are using. Is it SWI filtered phase or it's just a phase? And I think that clearly determines how much potentially abnormal phase you are going to see. We are using what Professor Hecky recommended, 64 by 64, and we created so-called MPATP, which stands for the mean phase of the abnormal phase tissue. This is again something similar that you just showed in the previous presentation, 2 standard deviation above the reference group that has been created on approximately 123 healthy controls in linear way and has been published in Neuroimage last year. Now, you can see that the most affected region in CIS patients is the pulvinar nucleus of thalamus with 0.04 value, but you can see that all regions, all of them, have higher abnormal phase, with some of them reaching even 12% and being significant putamen. So clearly we see a more abnormal phase in CIS patients at the first clinical attack than in healthy controls. In terms of, so we are not just, but once we define the abnormal phase, we can easily determine the volume. And so if you look the volume of the normal phase, it's about 20% higher in CIS patients than in healthy controls in the pulvinar nucleus of thalamus, and about 17% higher in putamen. And that's highly significant. But if you take total deep gray matter, all basal ganglia together, it's also significant, 6.1%. Now, we looked, and I'm not presenting you, actually I'm presenting you the normalized volume, so it's volume per each structure, and you cannot find that neither in one of these regions there is a significant difference. So there is no atrophy in those regions. And so the conclusion of this study, in our opinion, is that iron is preceding, or whatever you want to call it, abnormal phase, is preceding tissue-specific atrophy at the first clinical onset. My second study is, or our second study, is on importance of iron deposition and disability in MS. This paper is also in press now in Frontiers of Bioscience. 210 MS patients, 149 with relapsing remitting, and 61 with secondary progressive. As you can see, very well expected characteristics for RR and SP. The difference is all in right direction respect to the disability, T2, T1 lesion volume, cortical volume, and also white matter volume. When we look the SWI abnormal phase, you can see that basically we see the expected. In the total deep gray matter, there is a difference between RR and S patients, caudate, putamen, trend for pulvinar nucleus of thalamus. The same for the atrophy. You can see that atrophy is much more present at this stage of the disease. One thing that we found in all these studies is that we begin to lose a lot of iron on imaging as we lose a lot of atrophy, because as you lose at the first clinical attack, we find usually more abnormal phase, but later on a lot of this abnormal phase is lost due to atrophy on the images. So the key question in this study was how atrophy and iron of the deep gray matter is doing respect to the established MRI measures. And I think that nothing better than to study in 210 people with MS. And so you can see that when we put in the model disease duration and EDSS and iron or abnormal phase, atrophy, T2, T1 lesion volume, cortical volume, and white matter volume, these measures do not enter into the equation. The only measures that remain significant are the total deep gray matter of abnormal phase and total deep gray matter abnormal volume. When you force at any cost the T2, T1 lesion volume, cortical volume, and white matter volume in the model, they, even by being forced, they are negative, with iron remaining the only in the model explaining higher EDSS. Of the particular structures, we found that caudate, red nucleus, and amygdala had high volume. High abnormal phase. I would like to point out that this presentation is also a platform at American Academy of Neurology, and we have had two other posters, of which one is also platform at American Academy of Neurology on pediatric MS populations. And just final is to say that in these very early patients at CIS and in pediatric MS, we are finding a very close correlation between inflammation in terms of T2 lesions and gadolinium lesions and amount of abnormal phase in these structures. I would be happy to answer the questions, and I'm done ahead of time.
Thank you, Robert. This is very interesting data on CIS because you clearly demonstrated that iron deposition precedes atrophy of the structure you measured. Did you correlate the iron content with duplex evidence of CCSVI in those patients, in those CIS patients?
I mean, we are planning to do it, but I can't give you this data at the moment.
Great. Robert, thank you very much. I'd like to have you all know we're going to start again at 1 o'clock, and the lunch today may actually be back in the exhibitors hall so we can spend some time there. And please, we will start at 1 o'clock with the next session. Thank you.